Hello everyone, this video is about DNA replication and its key points. So, DNA replication key points. DNA is a molecule made up of two strands twisted around each other in a double helix shape. Each strand is made up of a sequence of four chemical bases represented by the letters A, C, and G and T. The two strands are complementary. This means that wherever there is a T in one strand, there will be an A in the opposite strand, and wherever there is a C, there will be a G in the other strand. Each strand has a 5' end and 3' end. Two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of the replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. Our DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps which are left behind with DNA. Finally, the enzyme DNA ligase scales up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old conserved strand of DNA and one new one. Then about DNA replication. DNA replication is a fundamental process occurring in all living organisms to copy their DNA. This process is called replication in the sense that each strands of dsDNA serve as a template for reproduction of complementary strands. General features of DNA replication. DNA replication is semi-conservative. It is bidirectional process, not unidirectional. Never forget that. It's a bidirectional process. It proceeds from a specific point called origin. It proceeds in 5' to 3' direction. It occurs with high degree of fidelity. It is a multi-enzymatic process. DNA replication is semi-conservative. It is bidirectional process. It proceeds from a specific point called origin. It processed in 5' to 3' direction. It occurs with high degree of fidelity. It is multi-enzymatic. It occurs with high degree of fidelity. That means mismatch repair will be happening. Repair processes. Okay. So it is a multi-enzymatic process. DNA replication occurs by 3 steps: initiation complex formation, closed complex formation, open complex formation. Once again, initiation complex formation, closed complex formation, open complex formation. So elongation, leading strand synthesis and lagging strand synthesis, termination, DNA replication in prokaryotes we are going to learn. Initiation. DNA replication begins from origin. In E. coli, replication origin is called RIC, which consists of 245 base pair and contains DNA sequences that are highly conserved among bacterial replication origin. Two types of conserved sequences are found at ORC: 3 repeats of 13 base pair and 4 to 5 repeats of 9 base pair. Okay. Then here we could see a picture of this. I will be zooming. Then 9 dimers, then 13 meres, negatively supercoiled template DNA, initial complex, open complex, ATP, preparing complex, pre-priming complex. About 20 molecules of DNA A protein binds with 9 mers repeats along with ATP, which causes DNA to wraps around DNA A protein, forming initial complex. The DNA A protein and ATP triggers the opening of 13 mer repeats, forming open complex. 2 copies of DNA B protein helicase binds to 13 mer repeats. This binding is facilitated by another molecule called DNA C. The DNA B DNA C interactions causes DNA B ring to open, which binds with each of the DNA strand. The hydrolysis of bound ATP releases DNA C, leaving the DNA B bound to the DNA strand. The binding of helicase is key step in replication initiation. DNA B migrates along the single-stranded DNA in 5' 3' direction, causing unwinding of the DNA. The activity of helicase causes the topological stress to the unwinded strand, forming supercoiled DNA. This stress is relieved by the DNA topoisomerase, DNA chirase. By negative supercoiling will be taking place. Similarly, single-stranded binding protein binds to the separated strand and prevents reannealing of separated strand and stabilizes the strand. The DNA polymerase cannot initiate DNA replication, so at first primates synthesize 10 plus or minus 1 nucleotide RNA in nature along the 5' 3' direction. In case of E. coli, primer synthesized by primase starts with PPP AG nucleotide. Primer is closely associated with the DNA B helicase, so that it is positioned to make RNA primer as SS DNA of lagging strand. Next is about elongation. Here we could see a picture which depicts this. The fork moves as replication takes place. 5' old strand, 3' old strand, 3' old strand and 5' old strand, 5' new strand, leading strand, then lagging strand. Replication fork. First is about leading strand synthesis. Leading strand synthesis is more straightforward process which begins with Leading strand synthesis is more a straightforward process which begins with the synthesis of RNA primer by primase at replication origin. DNA polymerase 3 then adds the nucleotides at 3' end. The leading strand synthesis then proceed continuously, keeping pace with unwinding of replication fork until it encounters the termination sequence. First is about leading strand synthesis. Leading strand synthesis is more a straightforward process which begins with the synthesis of RNA primer by primase at replication origin. DNA polymerase 3 then adds the nucleotides at 3' end. The leading strand synthesis then proceeds continuously, keeping pace with unwinding of replication fork until it enters the termination sequence. Then lagging strand synthesis will be taking place. The lagging strand synthesized in short fragments called Okazaki fragments. At first, DNA primer is synthesized by primase, and as leading strand DNA polymerase 3 binds to RNA primer and adds DNTPs. On this level, the synthesis of each Okazaki fragment seems straightforward, but the reality is quite complex. So, mechanism of lagging strand synthesis. Template DNA is there, RNA primer, lagging strand, SSB protein, single stranded binding protein, then DNA primase, DNA helicase, template DNA, leading strand, DNA polymerase 3, Okazaki fragment, 3' and 5' regions, 5', 3' parent strand, direction of fork movement, DNA polymerase 3. The complexity lies in the coordination of leading and lagging strand synthesis. Both the strands are synthesized by a single DNA polymerase 3 dimer, which accomplished the looping of template DNA of lagging strand synthesizing Okazaki fragments. Helicase DNA B and primase DNA G constitute a functional unit within a replication complex called primosome. DNA pol 3 use one set of core subunit, core polymerase, to synthesize leading strand and other set of core subunit to synthesize lagging strand. In elongation steps, the helicase in front of primase and pol 3 unwind the DNA at the replication fork and travels along lagging strand template along 5' to 3' direction. DNA G primase, occasionally associated with DNA B helicase, synthesizes short RNA primer. A new B sliding clamp is then positioned at the primer by B clamp loading complex of DNA pol 3. And the Okazaki fragment synthesis is completed, the replication halted, and the core subunit dissociates from their sliding clamps and associated with the new clamp. This initiates synthesis of new Okazaki fragments. Both leading and lagging strand are synthesized coordinately and simultaneously by a complex protein moving in the 5' to 3' direction. In this way, both leading and lagging strand can be replicated at the same time by a complex protein which moves in the same direction. Every so often, the lagging strand must dissociate from the replicosome and reposition itself so that replication can continue. Lagging strand synthesis is not complete until the DNA primer has been removed and the gap between adjacent Okazaki fragments is sealed. The RNA primer are removed by exonuclease activity 5' to 3' of DNA pol 1 and replaced by DNA. This gap is then sealed by DNA ligase using NAD as cofactor. So termination. Eventually, the two replication fork of circular E. coli chromosome met at termination recognizing sequence. Eventually, the two replication fork of circular E. coli chromosomes met at a termination recognizing sequence TER. The TER sequence of 23 BP are arranged on the chromosome to create a trap that the replication fork can enter but cannot leave. TER sequence functions as binding site for TUS protein. So origin is there, daughter strand is there, parental strand is there, replication forks are there. So replication proceeds in both directions. Origin, daughter strand, parental strand, replication forks. Replication proceeds in both directions. Termination of replication. TER-TUS complex can arrest the replication fork from only one direction. TER-TUS complex encounter first with either of the replication fork and halted, the other opposing replication fork halted when it collide with the first one. This seems the TER-TUS sequence is not essential for termination, but it may prevent over-replication by one fork if other is delayed or halted by a damage or some obstacle. When either of the fork encountered TER-TUS complex, replication halted. Final few hundred bases of DNA between these large protein complexes are replicated, but not yet known mechanism, forming two interlinked or catenated chromosomes. In E. coli, DNA topoisomerase 4 type 2 cut the two strands of one circular DNA and segregate each of the circular DNA and finally join the strand. The DNA finally transferred to two daughter cells. Then DNA replication in eukaryotes. DNA replication in eukaryotes occur only in S phase of cell cycle. However, pre-initiation occur in G1 phase. Due to sheer size of chromosome in eukaryotes, chromosome contains multiple origin of replication. ARS, autonomously replicating sequence. In case of yeast is origin for replication. Then steps in DNA replication. Initiation is the initial step. The first step in the formation of pre-initiation replication complex, pre-RC. It occurs in two stages. First stage required there is no CDK activities. It occurs in early G1 phase. It is known as licensing, but licensed pre-RC cannot initiate replication at G1 phase. Second stage is binding of ORC, origin recognition complex. The replication begins with binding of ORC to the origin. ORC is a hexamer of related protein and remains bounded even after DNA replication occurs. Furthermore, ORC is analog of prokaryotic DNA A protein. After binding of ORC to origin, CDC6, CDC18 and CDT L coordinate the loading of MEM, mini chromosome maintenance to origin. MEM complex is thought to be major eukaryotic helicase. After binding of MEM complex to pre-RC, CDTL get displaced. Then CDK phosphorylates MEM, which activates its helicase activity. Again CDK and CDK recruit another protein called CDC45, which then recruit all the DNA replicating proteins such that the origin get fired and replication begins. So topoisomerase is there, SSPs, pole delta, sliding clamp PCNA, then leading strand helicase SSBs, pole A primase, along with some lead sliding clamp PCNA. Okay, Saki fragments, DNA primer, lagging strand. Then is about elongation process. DNA polymerase delta synthesizes and adds DNTPs at 3' end of RNA primer. The leading and lagging strands are synthesized in the similar fashion as in prokaryotic DNA replication. Then along with that, termination process will be taking place. Termination, connection of discontinuous segments. Here we could see. Then at the end of DNA replication, the RNA primer are replaced by DNA by 5' to 3' exonuclease, and polymerase activity of DNA polymerase. Exonuclease activity of DNA polymerase removes the DNA primer, and polymerase activity adds DNTP at 3' OH and preceding the primer. In case of bacteria with circular genome, the replacement of DNA primer with DNA is not a problem because there is always a preceding 3' OH in a circular DNA. But in eukaryotic organism with linear DNA, there is a problem. When RNA primer at 5' end of daughter strand is removed, there is not a preceding 3' OH such that the DNA polymerase can use it to replay by DNA. So at 5' end of each daughter strand, there is a gap, missing DNA. This missing DNA causes loss of information contained in that region. This gap must be filled before next round of replication. For solving this end replication problem, studies have found that linear end of DNA called telomere has GC rich repeats. This sequence is known as telomere sequence. These repeats of telomere sequence is different among different organisms. Telomere in human cell consists of repeats of this shown like this. Each species has its own species-specific telomere repeats. These telomere sequence do not code anything, but it is essential to fill the gap in daughter strand and maintain the integrity of DNA. Then telomere replication, end replication problem in eukaryotic DNA. There is an enzyme found in eukaryotic cell called telomerase. Telomerase is a DNA polymerase, DNA-dependent DNA polymerase, which adds many copies of telomere sequence at 3' OH end of the template strand. Like other DNA polymerase, telomerase is also add deoxyribonucleotide at 3' OH end. Unlike other DNA polymerase, telomerase add DNA at 3' OH end of parent strand, not at the daughter strand. And also it synthesizes the same sequence over and over in the absence of template strand. So telomere template, hairpin created, DNA RNA primer added, hairpin extended, RNA primer removed, gap filled, hairpin removed, replicated telomere. First telomerase binds to 3' OH end of parent strand by hybridization between it's this kind of shown like this RNA sequence and TTGGGG DNA sequence, telomere sequence of tetrahymena. The telomerase adds TTG at 3' end of parent strand. After adding TTG sequence, telomeres translocates along 5' 3' end of parent strand. Now the telomerase adds this to 3' end by using its sequence. Again telomerase translocates and adds this sequence. This process is continued for many times. The parent strand become more longer than daughter strand. Now RNA polymerase synthesize RNA primer by copying the parent strand in 5' to 3' direction using telomere sequence as template. The DNA polymerase can now extend in primer in 5' 3' direction by adding deoxyribonucleotide to 3' end. The primer is now removed and it won't be replaced because it is an extra sequence added by copying telomere sequence. Finally, the integrity of daughter strand is maintained.